Thank you. Thanks for having us. I'm pleased to present a status update on Abeona. Some of you have heard from us in the past meeting at the Mesa. So as a public company, we have to basically remind everyone that some of the statements we're making are forward-looking statements. Abeona is a fully integrated cell and gene therapy company. We have our both research and development as well as the manufacturing, GMP manufacturing facility out of Cleveland primarily, with a smaller administrative office in New York City. We have both programs in cell therapy, being the main one in recessive dystrophic epidermolysis bullosa, which is a similar indication to that of fibrocell, which you just heard before, and also a whole host of AAV-based direct gene therapies for both CNS disorders, lysosomal storage disease with CNS manifestation, as well as some other indications, which I'll go over in a minute. Here's our more advanced clinical pipeline and also a little bit about a preclinical pipeline. So I mentioned before the EB program, Epidermolysis bullosa program, is nearing phase 3 pending some stability data on manufacturing, which I can comment in a bit. The next 4 programs, ABEO 102, 101, 202, and 201, are in lysosomal storage diseases. The first 2 ones are in Sanfilippo A and B, or mucopolysaccharidosis 3A and B, and the next 2 are in CLN, neuronal ceroid lipofuscinosis type 1, which is infantile, and type 3, which is the juvenile type. We have also announced late last year that we have early program in cystic fibrosis using one of our proprietary AIM capsids. We intend to move this into the IND throughout this year and then next year. And then we have a whole host of novel capsids, which are chimeric AAV capsids. These are also out of that group of capsids. There are a variety of other programs which I have not announced, but some of which will be in retinal disorders. So I'll start with our RDEB, recessive dystrophic epidermolysis bullosa. As you heard before, this is an ultra-rare disease. We estimate there's about 2,500 patients in the United States alone. We have multiple designations, as you can see here. Of curiosity, we have both breakthrough and RMAT, which tells a little bit of the history of the program. Started a while back before RMAT was even in place. This is a program we in-licensed from Stanford University that did much of the work up to date. We recently transferred the IND as we prepare to move into phase 3, and of course, they've taken over the manufacturing from Stanford. I won't spend too much time here. My colleague in fibrocell gave a very nice explanation. Just suffice to say, and you can see a scary-looking picture here, which feels scary for the patients and for the families that deal with RDEB. This sometimes can involve a good percentage of the body, anywhere from 10-20% and upwards to 80% of the body can be affected by wounds. Majority of these wounds actually tend to be chronic. Most wounds are larger than 25 centimeters squared of all types. And if you look at the chronic wounds, which are the most common one and most disabling ones, these tend to be on average about 100 centimeters squared. When we say chronic, these are wounds that don't heal, pretty much don't heal. They can be open sometimes for years. In fact, some of the patients that came to the Stanford trial had these wounds somewhere from 10 to 20 years of duration. So it's pretty dismal. With the more extensive involvement of the skin, some of these patients have significant systemic complications ranging from, of course, risk of infection, chronic malnutrition, inordinate metabolic demands, requiring tube feeding, as well as a variety of other conditions. And patients with RDEB tend to live usually into their 30s and 40s and often succumb to squamous cell carcinoma, which is a consequence probably from these open wounds and perpetual attempt at healing. So the program, Abeona, is slightly different from that of fibrocell and different from Crystal, which is another company developing products for RDEB. Ours is derived from skin, so it starts out with skin biopsy, which is a punch biopsy, standard in dermatology offices. brought into the Abeona facility. These keratinocytes are isolated and then transfected with a retrovirus with the functioning collagen 7 gene. These cells are then grown into a sheet, as you see here in the right lower quadrant. These sheets measure approximately 35 to 40 centimeters in area. And this is important, although it seems trivial. What we heard from fiber cells, they focus primarily on the smaller wounds up to 20-something centimeters. Maybe they're growing to larger wounds. The Stanford program, now Abeona program, focused on larger wounds, 20 centimeters or larger. In fact, some of these were hundreds of centimeters in area when treated. Once these sheets are ready, these are then transported to the site, and they are transplanted surgically. It's basically a simple grafting procedure. We can manufacture 6 of these sheets, which in total can cover an area approximately 210 to 240 centimeters square in the patient. So in effect, addressing the larger, most disabling wounds of these patients. So in some ways complementary to some of the other programs. The phase 1/2 trial, as I mentioned, was conducted by Stanford. These, again, patients who had very long-lasting, large, horrific-looking wounds. You saw one of the pictures. Here, patients were started in the phase 1/2 trial, all young adults. We have now ability to treat children 6 and older going forward into phase 3. These patients had multiple wounds treated, anywhere 5 to 6 wounds per patient. There were 7 patients enrolled, so I'll give you a snapshot of what the results were. These results were published in JAMA, and there's an upcoming follow-up of this with a 3-year follow-up mark from these patients. An accepted publication we'll announce when it comes out. What you can see here on the left side of this picture is one of the wound sites, and by wound site you see the delineation with these dots here, which are inked onto the skin. This is an area of 35 centimeters squared, which is essentially an area where the graft will be placed. So is a corrected keratinocyte, autologous gene-corrected keratinocyte sheet. And you see at 3 months fully healed, 6 and 12 months fully healed. And as you've seen before, and I'm showing here for our product, you see presence of collagen 7, as you see in the green fluorescent panels below. This particular patient was been followed now for 2 years. We have data actually out to 4 years in this very patient, and it continues to have healing long term. This is unprecedented in RDEB, in part because this patient specifically had wounds lasting 3 to 5 years before that had never healed, and now the wounds have remained healed for 4 years. This is the full heat map of all the 7 patients treated. There are different stages of follow-up. The first patient treated is now out to 5 years. Most wounds healed from this, except for patients number 4 and number 6, which are patients with 2 specific issues. Patient 4 was found to have pre-existing antibodies in hindsight, and that's something we're actually going to screen for going into phase 3. So that patient eventually had a graft rejection from pre-existing antibodies. It did heal for a while, but then it didn't last. And patient 6 was arguably the most difficult area to treat, as a patient had a massive wound of 400 centimeters square, essentially occupying the entire back. This patient, again, wanted this, obviously the most disabling wound he had, and the team at Stanford decided to treat it with 6 grafts, so covering approximately half of the wound size. And unfortunately, this eventually got infected, and that affected the graft stability. It did heal, surprisingly, even despite the being such a large wound. So going into phase 3, we obviously have had to transfer the manufacturing from Stanford to Abeona. So you've heard throughout the day, the manufacturing is not a trivial effort, but we've done this successfully. We've manufactured hundreds of these sheets repeatedly and inconsistently. And we're looking to do a phase 1/2 trial. We'll start at Stanford, which is this primary center, and we'll continue as a multi-center trial with other centers coming on board. We'll announce those as they're ready to go. We have agreed with the FDA that we will have, again, endpoints at 3 months for efficacy and 6 months for durability. And of course, these patients are followed longer term. Wound healing is the primary outcome measure. But different to some of the other programs that are looking at much smaller wounds, we are looking at a threshold that is less than 100%, because it will be unrealistic, especially in children, to expect that very large wounds, especially 20 to 40 centimeters square, that wouldn't necessarily have full healing. In fact, perhaps in the larger wounds, one better measure would be even the amount of skin that is healed, because from the point of view of the patients, they're less concerned about a percentage number. They're more concerned how much wound they have left open, which is really the source of main disability. The large wounds have excruciating pain. You've heard of a sickle cell disease example of people on chronic opioids. So are the people with large RDEB wounds. They often require chronic opioids and also acute painkillers just for the wound dressing changes that are done several times a week. So it's an excruciating disease. So switching gears to the AAV-based programs, these are AAV9, the first four programs are AAV9-based. These are licensed, the capsids are licensed from Regenxbio. The programs came from various universities, both Nationwide Children's Hospital, UNC, and also University of Nebraska. Patients, the first two diseases in mucopolysaccharidosis 3. I'm just going to be brief about this just because I'm running out of time soon. These are actually devastating disorders. There are systemic defects in the lysosomal enzyme that leads to accumulation of these sugars, main of which is heparan sulfate. Heparan sulfate accumulates in the lysosome, causes this dysfunction of lysosome, which leads to cell dysfunction and ultimately cell death. Whereas this is a systemic disorder with a variety of systemic abnormalities, including enlarged liver, coarse feces, and whatnot. The main disability of these patients is due to neurological impairment. They have a deceleration in their neurodevelopment, followed by plateauing and actually frank dementia starting, early in life. So by age 4 or 5, these children are quite disabled already. Primarily cognition, not as much motor. The programs are replacing the defective transgene with a normal functioning enzyme, and it's the same as for both programs. Actually, the approach itself is similar to both programs. We dosed patients in a phase 1/2 trial for MPS IIIA. This is a multi-center trial, multinational, in 3 dose cohorts. Currently dosing patients in cohort 3 with the 313 vector genomes per kilo. Primary outcome measure, in addition to safety, is cognition. We are, of course, measuring a variety of biomarkers, especially because in this disease we can follow the accumulation of the substrate for the defective enzyme. Nationwide Children's Hospital, University Women's and Children's Hospital in Adelaide, Australia, and the Santiago de Compostela in Spain are the main sites. Here's a snapshot of the by cohort, by dose cohort. So in purple is the low dose cohort, red mid-dose cohort, and blue highest dose cohort. You see a clear drop in heparan sulfate levels in the CSF, and this is over time. CSF heparan sulfate levels we believe to be a manifestation of this neurological disorder, as close to biomarkers you can get, short of course, a biopsy or direct examination of the tissue. And we also view this as a good proxy for evidence that this vector, which is delivered intravenously, is getting into the CNS, which of course you have evidence from the Vexus drug Zolgensma, which is also intravenously delivered for a CNS disorder. Likewise, you see a reduction in liver size. This is a biophysical measure measured by MRI. Here's just a multiple of the normal liver for age. Obviously, liver grows with age. So here you have a decrease, which is also dose dependent. And finally, the neurological outcome measure. This is in cognition by a scale called the Mullen Developmental Scale. The x-axis is months age in months, and the y-axis is the cognitive age equivalent, so essentially a measure of cognition. The background gray data points are the natural history data points for children who were untreated. This is out of the Nationwide Children's Hospital cohort. There are several other cohorts we have access to. These are public data. And you can see here the data are shown in color coded per cohort. The purple are the first dose cohort, lowest, also treated older patients who were more affected at the time of treatment. And they did not show a clear response in this particular outcome measure, although they did have improvement of their biomarkers, and the families also report some improvements which are not captured by this measure. The patients in red, 3 patients in cohort 2, are younger, treated mid-dose cohort, one of whom plateaued for 18 months, then had a slight decline in cognition. We'll see what happens over time. 2 others didn't seem to respond based on cognition. And then you have cohort 3, which are in blue, and it's sort of a range of ages. Treated patients are much older, but then also the 3 youngest patients in our clinical trial, these were treated under age 30 months. And these children all remain fairly close to the normal developmental range for these, lines. If you see this line written DQ100, this is developmental quotient, 100% of the normative data for this scale. In particular, if you see the patient who's the older patient who's been followed now for 18 months, that child continues to acquire skills and is attending school. And none of the patients that have been reported in natural history studies have ever reached this level of cognitive development. So this is a clear outlier. The other ones are younger, but they are attracting pretty nicely. And of course, we continue to follow these patients. So we believe that combined, both in terms of the clear biological evidence of efficacy and in the biomarkers and biophysical measures, as well as the early evidence of cognitive improvement, suggests that this is in fact an active, suggestion or show that this is a biologically active product that is now showing cognitive improvements. And not surprisingly, the evidence of effect is greatest in the youngest children. And this is something we've shown time and again on our own data, preclinical data, but also others have shown in mucopolysaccharidosis when early intervention has clear correlation with ultimate outcome. So here's the summary, which I already provided, and we actually engaging the agencies both in the US and Europe to discuss next steps for this program, both in terms of clinical development as well as manufacturing as we transition to a later stage program. The phase 1-2 program in MPS IIIB is earlier in that we had enrollment of a patient 2 years ago, and then most recently we enrolled an additional 5 patients to the program. So this patient population within the study is much shorter follow-up, but is built the same way, same approach, with the difference that this is not a self-complementary vector, so we have to start it out with a little higher dose, and we'll push to a dose even higher than this. Patients here also responded with an improvement of the accumulation of heparan sulfate in the CSF, as you see here in purple, and then also in the urine. On the right panel and the left panel is liver size, and you see in gray background, these are natural history data set from children who were not treated, and they continue to have large livers over the age. Here's cognition. We have 1 data point in the older patient. We're accumulating additional points, but most of these patients were enrolled over the past several months, so we don't have sufficient data on the cohorts and the various cohorts. So switching to Batten disease, which is also lysosomal storage disease, this disease, in particular the infantile form, affects younger children, presents a slightly different, although equally devastating disease, often with epileptic seizures early in life. These children also have visual loss, so they become blind as the disease progresses, and also they have neurocognitive impairment and ultimately decline and die earlier than people with Sanfilippo syndrome. Here, this program was done in collaboration with the UT Southwestern, Dr. Stephen Gray, who's been a researcher in this area. Also AAV9-based program replacing the defective enzyme, PPT1. What's different about this program, in part because of the aggressive nature of the disease and also the pathology that it not only involves the brain but other areas of the CNS, including the spinal cord, is that we are approaching a combined route of delivery, both intravenously and intrathecally. So this program had the FDA has given us go-ahead earlier this year to move into phase 1, so we're just planning to start the phase 1-2 trial in the coming months. So lastly, we have a very active program of the next generation AAV vector platform. This was originally out of the University of North Carolina. We licensed in a large library of novel capsids, which we have continued to evolve within the Abeona Research and Development Facility, now extended out to 100 different capsids, including some novel IP. Essentially a reshuffling of the wild serotypes of AAV, leading to capsids that could potentially have better tropism to different organs. And also, as we heard earlier, especially in patients with either pre-existing immunity to AAV or who've been previously dosed with an AAV-based vector, these novel capsids could potentially evade immunity, which is obviously an advantage both for redosing or dosing children with already existing immunity. So we have already been able to provide, to generate proof of concept activity with these actual capsids in a variety of animal models in multiple disease states, and these have been presented or hopefully published at some point. Last, and just maybe going back, we always make the same kind of quasi-joke. This is not stock photo. This is actually from our manufacturing facility in Cleveland. These are real people who work there. We have a fully integrated manufacturing, a GMP manufacturing facility in Cleveland that has 6 GMP suites. 3 are now dedicated for EB101, which is the cell therapy program, and could support commercial launch and treat up to 120 patients in the first year. Obviously, it wouldn't require expansion further to accommodate the rest of the population. And the other 3 are also CGMP units for AAV manufacturing. We have manufactured routinely our own vectors for all the research activities, and we're now moving forward already with some experience out to 160 liter, 4 times 40 liter reactor suspension HEC-293 cell production. So we're on our way to supplying our own programs with manufacturing out of Cleveland. Very proud of this work, and that it obviously required a tremendous amount of investment, not only in terms of financial investments, but also staff has done a terrific job, but also the quality systems that cover both the AAV facility as well as the EB101 facility. So that's actually an advantage. And I'll stop here. Thank you.